Good morning and welcome everyone, especially our respected principal sir, respected research person, delegates, participants, and dear students. We are going to start the national webinar on application of bioinformatics, a case study on sequence analysis, phylogeny, and protein modeling of novel coronavirus. This webinar is organized by the Department of Zoology of Dhing College. We are fortunate to have Dr. Chittaranjan Barua, well-known herpetologist in Northeast India, and assistant professor senior grade in Durham College as a research person. Protein structure prediction is one of the most important goals pursued by bioinformatics and theoretical chemistry. It is highly important in medicine, for example, in drug design, and biotechnology, for example, in the design of novel enzymes. The drastic increase in the number of coronavirus discovered and coronavirus genome being sequenced have given us an unprecedented opportunity to perform genomics and bioinformatics analysis on this family of viruses. Today's webinar is very important for drug designing of novel coronavirus, which is a very important goal for today's researchers, students, participants, and every citizens of the world during this pandemic season. Dr. Chittaranjan Barua is very experienced in this field, and he has published a lot of articles and papers in various international journals. We hope and believe that all the participants will be benefited his experience. Now I request our principal sir, Dr. Biman Hazarika, to deliver his inaugural speech and inaugurate this webinar. Thank you.
Thank you. Thank you, Dr. Luna Fupan, HOD, Department of Zoology in College. Good morning. Good morning, everyone. Today, Department of Zoology in College is hosting a webinar, national webinar, on a very special topic, that is bioinformatics, a case study on sequence analysis of phylogeny and protein modeling based on novel coronavirus. So, if we look into the topic of this webinar, then we will find that new things will come up and how our scientists and researchers are working in this pandemic situations to cope with this dreaded virus, which already we have lost a lot of people all around the globe. In this particular webinar, we have invited a very special person, an academician, a professor of Durham College. Currently, he is in the PG department of Durham College, Dr. Chittaranjan Barua. And I find, while I am going through his CV, I find it really awesome that he has done a lot in his own field and adding new things to his secured research career. He has, to his credit, UGC RF SMS fellowship, and he has already obtained awards from Asian Scholars Wetland Institution, and he has won a particular award in this context, winner of Conservation Leadership Award. And apart from his teaching, what I see and what others people see and observe that he has done a lot in his field. So far, he has, to his credit, 30 international and national seminar papers, and he has the experience of working in turtle conservation program inside and outside India. So today we are very lucky enough, particularly our students and esteemed participants will definitely enlighten with his speech particularly his research-oriented work, what he has done and what he is pursuing right now. And I am not going to talk about the bioinformatics system, which has done a lot, particularly in the field of medicine. Bioinformatics has contributed a lot. It helps a lot in biomedicine sector, in genetic compounding sector, and in other fields also. It helps a lot in generating different type of medicines, different type of components which are required in aiding in the field of medicine particularly. So I am not going in details to this particular field since I am not from the field of science. I belong to humanities. So leaving it to Dr. Borua, I would like to shed few lights on our institution. Dhing College is located in the northwest part of Nagaon district. Its catchment area is very big, 30 kilometers. It covers 3 assembly constituency segments, that is Dhing itself, Kotadroba, and Lahorighat. So we have 3 assembly segments in our catchment area. Our campus is having 75 bighas of land, and already 40 to 45 bighas of land being occupied by our departments, gymnastic hall, our sports ground, library, auditorium, etc. And despite that, we have 3 streams so far, newly introduced commerce as well. And we are going to offer, and so far we have been pursuing and offering for our students, targeting our students, skill development courses, and other such courses which are beneficial to our students who are pursuing higher education in our institution. So this is the college which was founded in 1965, and with the blessings of great Guru Shankardeva. Bordua is very nearer to us. So with the blessings of Mahapurush Srimanta Shankardeva and our forefathers, our founders, we are running our institution, and so far we have obtained B++ grade, and we are hoping for a new venture. We are planning to do something in the field of undergraduate education in near future. And with this word, I must congratulate, I must extend my heartfelt thanks to the department fellow, particularly the Department of Zoology, who is now doing a lot in this field of organizing this type of webinar, focusing on new lights, on new aspects, new issues. I feel really proud of them. I extend my heartfelt thanks to Dr. Luna Fupon, HOD, Department of Zoology, Dr. Purnima Das Bora, Associate Professor of the same department, along with their junior fellow, junior colleague, Dr. Ujayendra Das, Assistant Professor, Department of Zoology. I believe our department, various departments, particularly I'm talking about our science department. You know, in the field of science, so many things can taken up by our department fellows. So particularly, I request our department fellows, particularly the Department of Zoology, to follow what actually Sitaranjan Borua is now doing in his own college. If we follow him, our particularly our student friends will follow, then definitely they will be highlighted, they will be enlightened, they will be benefited out of that. So many research projects he has done. So I am offering this particular forum, webinar forum, to him particularly, Dr. Borua, and I wish a very successful webinar. in this hour of morning hour. And I also extend my blessings and wishes to all of you for forthcoming Durga Puja. With these words, on the desk of principal, I congratulate all and wishing you all a very successful webinar on this topic, bioinformatics. Thank you.
Thank you, Dr. Hazarika sir, for your valuable suggestion. Now, allow me to introduce our venerated research person, Dr. Chittaranjan Borua. He is currently Assistant Professor, Senior Grade, Department of Zoology, Durham College. He is the founder, editor, and board member of 10 international journals and assessor of 6 national journals. He has published 26 international and 10 national journal research papers. He published a book on turtle conservation of India. He attended more than 30 international and national seminars. He worked as a research person at least 10 different higher educational institutions on bioinformatics. Besides this, he was awarded and got fellowship more than 8 times nationally and internationally, and he completed 8 professional research experience both nationally and internationally. He is the editor of a UGC journal. He worked together in many international scientists, namely M.A. Laskar, head of the biotechnology, St. Anthony College, Shillong, Dr. Kartik Shankar, IS Bangalore, Dr. Osuk Kumar Malik, Dr. Shailendra Singh, and renowned herpetologist A.H.M. Ali Raga in Delta State University, USA. He is a life member of IUCN from 2009. Again, I welcome Dr. Borua to start this session. Thank you. Dr. Borua.
Thank you, ma'am. Respected Principal, Dhing College, Dr. Biman Hazarika sir, respected Head of the Department of Zoology, Dhing College, Dr. Luna Phukon, all respected faculty members of Dhing College, and respected audience and dear participants. So I'm going to start with the topic. Actually, with the help of Madam, I have switched the topic as I'm working in this field of bioinformatics and doing some preliminary work on COVID-19 also. So I have switched this topic, but most of the slides in this presentation are mostly focused on for the students of undergraduate level, although some of the higher level also there. So my presentation will include 4 different parts. First of all, I will give a brief introduction on bioinformatics for the undergraduate students. Again, to understand the link of how bioinformatics is related to other biological science and biological data. That means starting from DNA isolation to the bioinformatics. That means from dry lab, wet lab to dry lab. Again, application of bioinformatics in different fields. And lastly, in the last part, I will be covering some of the case studies on ongoing COVID-19 using bioinformatics tools and techniques. So before going to start with the topic, many of the students might not be familiar with the bioinformatics. So what is bioinformatics? Before going to the topic, so we should know what is bioinformatics. So bioinformatics is the application of computer in the domain of life science. It is the field of science in which biology, computer science, information technology are merged into a single discipline. That means bioinformatics is a interdisciplinary subject. That means different disciplines are combined to form a new combination of subjects that is called bioinformatics. So bioinformatics is known by different names in different fields because it has different application in different area. It is also known as in silico biology. Since computers are made up of silicon chips, the bioinformatics is popularly known as in silico biology or computational biology. And in different domain of life science, it is known as by different name. In chemistry, while people used to use bioinformatics for drug designing, that part is known as chemoinformatics. Again, bioinformatics is used in ecology also for ecosystem modeling. that branch is known as ecoinformatics. If it is used in immunology, the immunology scientists name it as immunoinformatics. Likewise, in pharmacology, in drug designing part, scientists are using it with the name of pharmacoinformatics. Likewise, it has different names in the different fields. But bioinformatics mainly concerned with the acquisition, storage, analysis, and dissemination of biological data, because biological data are very huge, from sequencing to different ecosystem level. So what is done in bioinformatics? Actually, bioinformatics is having two different parts mainly. One is computation part and one is application part. So people with the knowledge of biology like us, we are mostly doing the application part, but those people who are having higher knowledge with the algorithm development, mathematics and statistical knowledge or information technology knowledge, those people are having good background with the software handling. So they will start with the development of software, different software, whatever we are using in the application part. That means computation part involves the development of algorithms, again software development, and different database construction. We are having different database. That means starting from biodiversity to the genome, we are having different types of databases where bioinformatics is most widely used. So that means bioinformatics, people from different field can work in the field of bioinformatics. That means starting from information technology, mathematics, statistics, and biological science, biotechnology. So most of the people can integrate their, contribute their own way in the field of bioinformatics. Regarding application part, most of the people from biotechnology and life sciences, that means zoology, botany, biochemistry, food technology, they are involved with the sequence analysis, functional analysis, and structural analysis using the software developed by the experts in the field of mathematics and computer science. So the sequence database searching. So once we have a new sequence, we have to search with the database of different of the same type of database. We have to search whether same sequence is available in different databases, whether our sequence is similar to some other sequences. Again, sequence alignment. That means there is a analysis called sequence alignment. That means sequence alignment is to looking the residue correspondence, whether our sequence of nucleotide or protein will match with any other sequences. That is called sequence alignment. There are different types of alignment. In higher level, we have local level alignment, global alignment. We have, if we compare two sequences, that is called pairwise sequence alignment. If we are comparing more than two sequences at a time, that is called multiple sequence alignment. Again, genome comparison. So genome comparison is one of the most important application of bioinformatics. So now people are comparing the different genomes of different animals after sequencing the different genome projects. So we'll come to that in the subsequent slides. Again, another application is gene and promoter prediction. That means any of the sequences might not have any DNA sequence might not have the genes. DNA means not the genes, but all genes are DNA. Because genes are the small part, functional part of the DNA. So there are exons, there are introns. Genes are mostly located in the coding genes are mostly located in the exon parts of the DNA. So again, another application is motif discovery. Motif is folding of the polypeptide chain in the super secondary structures. That means we have to see how our new protein are folded. Again, another application is the phylogeny. So phylogeny will come in details in the subsequent slides. Phylogeny is the study of evolutionary relationship. Such type of question, for example, whether human beings are evolved from the monkey, or whether such coronavirus is related to any other viruses. Such type of questions can be answered with the help of phylogenetic analysis. Again, coming to the functional analysis, one of the applications is the gene expression profiling. In each and every multicellular organism, we have developed from a single zygote. So each and every cells are having same genes. That means whatever genes are available in our heart cell, same are available in the lung cell or kidney cell. But all genes are not expressed everywhere. For example, insulin is present in each and every cells of our body. But insulin gene is expressed only in the islets of Langerhans or pancreas. So which genes are expressed, which are not, those can be studied with the help of different modern techniques. Like one of the technique is called serial analysis of gene expression. Another technique is called DNA microarray or protein microarray. So those microarrays are studied with the help of bioinformatics only because last part is involved with the handling of computer and software. Again, metabolic pathway modeling, starting from glycolysis, Krebs cycle, to there are thousands of metabolic pathways in our body for performing functions. So those metabolic pathways can be modeled and studied with the help of bioinformatics softwares. Again, protein interaction prediction. So this is a very promising area. Whether that area is known as docking. Whether a protein will interact with the another protein. That means, for example, hemoglobin. Hemoglobin is a quaternary structure where 4 different chains, 2 different alpha chains and 2 different beta chains are combined to form a quaternary structure. How those 4 chains are interacted, or how DNA binding protein histone is binding to the DNA. That means protein interaction means protein ligand or protein DNA interaction can be studied with another area called docking. Again, protein subcellular localization prediction. So this helps to understand the different cellular aspects, where is the actual location of the protein after production of the protein, where it will be located, where it will be subcellular. That means that is called subcellular localization of the protein that can be predicted. Again, coming to the third one, that is structural analysis. So nucleic acid structure prediction. Nucleic acid means DNA and RNA. So DNA and RNA is having its tertiary structure. That means to perform a function, any molecule must be folded into a stable tertiary structure. Then only function is possible. 3-dimensional structure for functioning. So protein structure. Secondly, protein structure prediction. So protein structure prediction is one of the promising area of bioinformatics. For example, in this, we will come to the subsequent slide. In the case of novel coronavirus, there are around 29 different types of proteins from coronaviruses, but out of which only around 12 structures are so far experimentally determined with the help of X-ray crystallography or NMR or cryo-electron microscopy technique. Then what about other proteins? So research cannot be stopped. So in the absence of experimental structure, with the help of bioinformatics, we can model a protein with the help of computer software, which may be sometimes up to X-ray accuracy. That means it is already well established that protein structure predicted using bioinformatics can be used as an X-ray structure in the absence of X-ray structure. So protein structure prediction is one of the promising application of the bioinformatics. Again, protein classification. Proteins are classified various on the basis of various ways. On the basis of domains also, proteins can be classified. Domain means a domain is a polypeptide chain or part of the polypeptide chain, which can fold independently to a stable tertiary structure. So proteins are classified on the basis of domains like alpha domain protein, beta domain protein, and alpha beta domain protein. That means a alpha domain protein contains only alpha helices in the protein secondary structure. For that, we must know what is protein structure. We'll come to that also something up to some extent. So protein structure means there are different level of protein structure. Primary structure, that means polypeptide chain is the primary structure of the protein. Then secondary structure, alpha helix and beta sheets. Again, tertiary structure, folding of the alpha helix and beta sheet into the stable 3-dimensional folding. Again, quaternary structure like hemoglobin, 2 different polypeptide chains are involved in the quaternary structure. So different bonds are involved, starting from peptide bond in two different amino acids, between 2 different amino acids in a polypeptide chain. Then hydrogen bonding in the case of secondary structure. In the hydrogen bond stabilizes the alpha helix and beta sheets. Again, there are disulfide linkage in the protein. Again, there are different hydrophobic electrostatic interactions. There van der Waals forces are there in the tertiary structure. So those can be protein can be classified on the basis of their alpha domain protein, beta domain protein, and alpha beta domain protein. So alpha domain protein example, best example is the hemoglobin, myoglobin, or beta domain protein are mostly the viral code proteins. Again, alpha beta domain protein, most of the enzymes they are of the alpha beta domain in nature, where both alpha helix and beta sheets are present. So proteins are classified on the basis of that. So bioinformatics is helping how many alpha helices are there in a protein, given protein, how many beta sheets are there, whether it is absent, or what are different terms in a protein. So those can be compared with the help of bioinformatics. Again, protein structure comparison, different protein structure. Suppose we have a new protein structure, whether that protein structure is having same fold, whether SARS coronavirus particular protein is having same fold, SARS-CoV-2 is having same fold with the old SARS coronavirus or Middle East respiratory syndrome coronavirus, whether they are same or different. Those can be determined only with the help of bioinformatics. Again, it is very difficult to draw a protein structure because it is three-dimensional. So it can be rotated with the help of software. We can see by rotating the software, we can see each and every part of the protein for comparison of the protein. So bioinformatics is helping us tremendously in various ways in different fields. So if you talk about since most of the people are from biology background here, so bioinformatics in the application in the field of biological science, if you think we can identify a new species with the help of bioinformatics. If someone is giving a nucleotide sequence using different computer program like BLAST or FASTA, we can identify a new organism. Or population genetics, whether the same species of Brahmaputra river are same in the Ganga river. So that can be studied with the help of population genetics in the using bioinformatics, because there is also in the same species there are sequence differences. That means gene and there is a concept called gene and environment. So different genes are expressed in different environment. Again, coming to the sequence analysis, biostatistical analysis, and PCR primary designing. So PCR will come in details in next slides. So those things can be done with the help of bioinformatics. For PCR, we need primer, forward and reverse primer in general PCR. So those can be designed in the absence of a primer. Those can be designed with the help of software, which will reduce the experimental cost and time. Again, phylogenetic analysis and phylogeography. Phylogenetic analysis can be done to see the evolutionary profile of various organisms, and phylogeography, distribution of animal along with the phylogeny, can be done with the help of phylogeography. Again, coming to the molecular modeling and molecular marker of species. Different markers are there, will come in details in subsequent slide. There are different markers for species identification. That means suppose in airport, different airport will sometimes get the news. In different airports, different animals are rescued or their body parts are detected in the airport. So where will send those animals? If they are live specimen, where will send those specimens? Because suppose for one animal of Kaziranga, we cannot transfer that animal to other part of the country, because that's different area is having different animals of different area are having the different genetic setup. So there will not be purity of the genetic material otherwise. So unless and until we will know the specific location of the animal, that animal should be legally sent to the zoo only. Otherwise, if you know the location, then we can send, if a turtle species is recovered in the Delhi airport, if you know, have some study of, suppose from Kaziranga, if that species is detected from Kaziranga, then we can send it to their natural habitat. Otherwise, it is not possible. So molecular marker or species identification helps us to determine the exact location of the species. That means not only the species, from where is the species, that can be done with the help of bioinformatics. Again, biological database and species management. There are different thousands of biological databases. Whatever we are the database content the required information. Sequence database content sequence information. So we will come to that also in subsequent slides. Again, some other applications like genomics and proteomics, metabolic pathways, drug designing, and pharmacogenomics. So those are pharmacogenomics is one of the promising area of pharmacology, where pharmacology and genetics are merged into a single discipline, pharmacogenomics. That means pharmacogenomics deals with the individually tailored medicine. In developing country like us, we are using same medicine. Whatever medicine our parents are using for some type of disease, common disease, same type of medicine we are using each and every person in the community. But in advanced countries like US, what they are doing now. They are having genetic studies. They are having genome data of their own, and for some life-threatening disease like cancer, they are using personalized medicine. That means individually tailored medicine. That means drugs are designed based on individual's genetic profile. So that means মোৰ কাৰণে যিটো ঔষধ থাকিব সেইটো মইহে ব্যৱহাৰ কৰিম, বেলেগে নকৰে. So that is called personal medicine called pharmacogenomics. That branch is called pharmacogenomics. So how bioinformatics is related to biological data? As you know, our biological information is from, if you think about the defined level, defined molecules are there for biology. For animals, we are having the defined molecules, like we have protein molecule, we have DNA, we have carbohydrate, we have lipid molecules. So how those molecules are constituted? Those are made up of atoms, defined atoms. Carbon, if we were talking about DNA is having phosphorus also, nitrogenous base, then sugars. Again, protein is also having, apart from carbon, hydrogen, and other things, it is having sulfur also. So that means at lowest level of the biological study is the atomic level, then is the molecular level. Defined molecules are combined to form the cell, and different cells will form the multicellular, if it is a multicellular organism. Different cells will form the organism, and at higher level, it is from the species and more so on. That means biological study can be from different level, from physical level, chemical level, biological level, physiological level, and ecological level. That means we have given the scope of study. One who are interested to study only with the atoms, they can study with the different atoms. That means nanobiotechnology, they can deal with the help of bioinformatics. People who are interested to study different proteins and other molecules, DNA macromolecules, that at chemical level, they can give input with the help of bioinformatics and other life science subjects. Again, at biological level, different cells can be studied, and organism level, different physiological events of the organism can be studied with the help of bioinformatics. And there are nowadays some softwares also for study different. If you take a medicine, what will happen to our body? There are some software called ADMET, absorption, digestion, metabolism, excretion, and toxicity. That means software will tell what will happen if that medicine will go inside our digestive system. What will be the side effect and all. So all the data are incorporated into the computer, and the computer will tell what will happen to that medicine after incorporation to the digestive system. Again, ecological level, people are using with the various ecological studies, various ecological indices are there, and various population genetic studies and all. So people are using in different level. People are, apart from other different branches of biology and other tools, people are also using biology. For example, ecosystem modeling. People are using bioinformatics. For example, so what will happen to Kaziranga after 50 years? So those can be predicted with the help of like active prediction. Those can be predicted. Suppose what will happen if rhino is eliminated from some keystone species is eliminated from a particular habitat? So what will happen to that? So different data are incorporated into the computer, starting from rainfall data to climate change to different other aspects. So those by analyzing those data, so ecosystem can be modeled. So people are so developing different ecosystems with the help of this concept, with the help of ecosystem modeling using bioinformatics. That means biological information is from atom to species to ecosystem level. And we have whatever we are doing nothing. We are doing gene expression. And different body colors, different eye colors, those are involved with the expression of the genes. So how this class is given only because of understanding how bioinformatics is correlated with the molecular level informations. That means this, starting from the DNA isolation, we have to do the things for molecular biology works on bioinformatics. So for understanding, we have to know what are the basic understanding, what are the genotype, what are the phenotypes, because for each expression behind there is a gene. If there is a green color eye, for behind that there is a gene expressed for that. That means whatever we are doing in our day-to-day life. We are doing nothing. We are doing gene expression, different genes are expressed. Because if you think about different, we'll come to that. So for bioinformatics, the basic concept behind the bioinformatics application in the field of life science is the central dogma of molecular biology. That means once we have the DNA sequence, we can translate it to mRNA sequence, or subsequently we can get the protein. If somebody is giving me the DNA sequence, I will give him the protein sequence with the help of this concept. That means the central dogma of molecular biology is the basic principle of the working with the bioinformatics. So only because on the basis of central dogma and reverse, there is reverse central dogma also. In case of, for example, coronavirus, you can think the RNA is the genetic material in SARS coronavirus. So where RNA is first reverse transcribed to the DNA, single stranded DNA, with the help of reverse transcriptase enzyme. Then only from DNA mRNA is produced. That means to produce a protein, there must be one mRNA molecule. Other RNA cannot produce protein. There are different types of RNA. There are more than 64 different species of RNA. We know about basic RNA, mRNA, tRNA, and rRNA. So we know about different types of tRNA. For different proteins, for 20 different amino acids, there are 20 different tRNAs. So more than 64 different types of RNA are there, out of which only mRNA will give rise to the protein molecule. That means in bioinformatics, if somebody is giving us the RNA sequence, we can simply replace the RNA means what? RNA sequence means AUGC sequence, adenine, uracil, thymine, and guanine. DNA means ATGC, adenine, thymine, guanine, and cytosine. If DNA to RNA means one strand of DNA will be T will be replaced by U. So that is the mRNA. So with the help of bioinformatics, there are lots of softwares, there are lots of tools which will help us to translate a DNA sequence. That means DNA to RNA, RNA to protein we can do. That means from protein also, if somebody is giving us the protein sequence, we may know what was the DNA sequence for that. So on the basis of that, different types of analysis can be performed with the help of bioinformatics, with the concept of central dogma of molecular biology. We also know that there are different genetic code tables. This is just whatever we are getting in our conventional book, that is a standard genetic code table. But apart from this, there are vertebrate mitochondrial genetic code table, yeast mitochondrial genetic code table. And we know that protein sequence means 20 different L-alpha amino acids are written in the capital letters. So those are, suppose G for glycine, L for alanine. Likewise, one letter code, capital letter, that is protein. G for glycine, L for alanine, I for isoleucine, M for methionine, like that. For DNA means we know that 4-letter code ATGC nucleotides. So those will give rise to the different codons or different amino acids. So these are the basic knowledge before going to the bioinformatics application in the field of molecular biology. Unless and until we know this basic concept, it is very difficult to use bioinformatics in the field of life science. We also know that our body is made up of different proteins. Different proteins. Protein means proteins are made up of from L-alpha amino acid. Because amino acid, there are more than 400 amino acids in nature, out of which there are two types of amino acid, D-alpha and L-alpha. Only 20 L-alpha amino acids. There are some rare amino acids also, like selenocysteine. But most commonly, 20 L-alpha amino acids give rise to the protein molecules, the building block of protein. So whatever we are doing nothing. We are doing gene expression of proteins. Starting from beginning of the day, we are doing nothing. We are doing gene expression. Whatever we are taking food, starting from ptyalin or salivary amylase in our mouth, in stomach, we have different enzymes like pepsin, renin. We have different enzymes in intestine, trypsin, chymotrypsin. All are nothing. All are but protein molecules. All enzymes in our body, whatever functions are performing in our body by the enzymes are protein molecule, except ribosome. Ribosome is a RNA particle. Except ribosome, all enzymes are protein. Likewise, different hormones. We are having three different kinds of hormone: peptide hormone, protein hormone, and steroid hormones. So peptide and protein hormones, we can think about those are nothing but amino acids or proteins, which will help us in performing various functions. So that means proteins are the main workhouse of the cell, and which are controlled by the genetic material DNA. DNA is controlling what will be the protein molecule, what will be the protein form in our body. For example, we can take the example of the hemoglobin molecule. So you can see the hemoglobin molecule is having its primary structure sequence. You can see in the second column, you can see sequence is there. The sequence is folded into 4 different polypeptide chains. 4 different polypeptide chains will integrate to form the quaternary structure. In the bottom corner, black color, the colorful protein you can see. So that is the quaternary structure of hemoglobin having 4 different ion centers, which is functional. And that is performing our function. So you can see hemoglobin is carrying different gases in our main RBC. Main hemoglobin is the main protein, which is taking key role. Without hemoglobin, we cannot think about our life because we are respiring only because of hemoglobin. So that means proteins are vital, performing vital function in our body. So for understanding how hemoglobin is involved in different functions, for understanding that, we must know the structure of hemoglobin, we must know the active site of hemoglobin, then only the function, proper understanding of the function is possible. That means the protein is having, each protein is having tertiary structure. That tertiary structure or quaternary structure must be understood to an active site must be recognized to know the actual function of the protein. So integrating these changes in the molecular biology, how molecular biology experiments are integrated to the bioinformatics research, starting from the DNA isolation to sequencing and sequencing to the bioinformatics. So I will be highlighting you with the some of the slides. DNA can be isolated. Most of us know that DNA can be isolated from any of the body parts, any of the material. For example, if it is a rhinoceros or if it is a tiger, we can do non-invasive technique. The excreta contain DNA. Rhino dung or elephant dung contain the DNA. Likewise, there is called scat analysis. Carnivores, excreta of carnivores is known as scat. Scat also can be collected for, that means for tiger, you can do scat analysis. Scat can be collected and the DNA can be extracted. That means DNA can be extracted from different parts. And there are different standard protocol for collection of samples of the DNA in the wet lab. Blood sample, there is standard protocol, mostly EDTA is used, heparin or EDTA, anticoagulant is used for collection. Likewise, tissue sample, mostly we use 70 to 95% ethanol for collection of tissue sample before processing for the DNA isolation. Likewise, hair sample can be collected in different container, in zip pouch, or fecal matter can be also collected in different container using standard protocol. After that, we must subject the samples for DNA isolation. Most popular method, there are different methods for DNA isolation, but most common methods are people are using in the laboratory are standard phenol chloroform technique, Sembrook and Russell. Again, chloroform octanol method are there. There are other methods also there. C-tape method, so there for DNA isolation. So using appropriate method, we have to isolate the DNA. After isolation of DNA, so DNA must be that DNA. That means DNA isolated means that DNA contain all the genes, each and every genes. That is the total genetic material, total DNA. And if you are interested for particular gene, we have to develop specific primer for that gene. And after developing specific primer, we will, suppose we are interested for insulin, we have to design insulin specific primer. We have to run. There is a machine called PCR, polymerase chain reaction. PCR machine. In PCR machine, we will amplify the DNA of interest. For suppose, for example, insulin. Then we will get the pure form of insulin in the polymerase chain reaction at required quantities. PCR is a technique which was developed in 1980 by Gary Mullis. For which he got Nobel Prize in 1992. So polymerase chain reaction is one of the most wide application. Now people are using RT-PCR for COVID-19 test also. It is used as a technique for molecular diagnosis, where computer is also helping in the advanced PCR for detection of the samples. So PCR involves various steps. I will not go in details, but after PCR, we can from one microliter. Only 1 microliter means 1 microliter means 1 by 1000 of a ml. That means 1 ml of 1000 1 microliter. From 1 microliter of DNA, we can produce if you run about 36 cycle of PCR, we can produce around 68 billion copies of DNA, 2 to the power 36. That means one cycle means we can, from one DNA, we will produce 2 DNA. Second cycle means the produced 2 DNA will participate. That means 2 DNA will participate to form 4 DNA. In the third cycle, that means 2 to the power n from that. That means 8 cycle means 2 to the power 8. 36 cycle means 2 to the power 36. That means 68 billion copies of DNA can be produced in around 3 hours. Within 3 hours, that means if you are having sample for viruses, we have little sample. We can increase the number of. That means like our Xerox machine, we can make Xerox copy of the particular gene of interest in a PCR machine. That means PCR is like one kind of Xerox machine where we can produce desired quantities or desired number of DNA or RNA. So for there are different things if you are thinking about the molecular marker development for identification of species, we have to think about the gene of interest. We may think different part like D loop region, mitochondrial D loop region, or cytochrome oxidase subunit 1. So there are different genes for different markers. Which marker means what? That marker will help to identify, like marker in our scale. That marker help us to identify how many feet are there for length. So likewise, this marker also help us to find out the exact individual. That means there are different markers. Good marker means that marker must be marker must be reproducible. That means same result must be obtained using same procedure in the same in defined condition. That means in defined temperature. That means if we are doing same experiment in test pool, same experiment in Dhing should give same result, or same experiment in high altitude in the cold temperature or in Shillong must give same result. Is it not same result? That is not good marker. That means same procedure when repeated in different parts of the world or different temperature must give the same result. So good marker will help us to identify the appropriate species at any condition. So those things can help us to for germplasm characterization, genetic diagnosis, characterization of the transformants, study of genome. So there are different aspects are there for marker development, where bioinformatics also help us in various ways. So commonly used genetic markers are, you can see RFLP, restriction fragment, length polymorphism. That means different genetic material in the different tubes are treated with the same type 2 restriction enzymes. R1, that means type 2 restriction enzymes are used in genetic engineering. Those restriction enzymes are treated, same restriction enzyme treatments are done in different species. For example, different species of honeybee, suppose we are doing RFLP. We have to isolate the DNA for different species of honeybee. For each individual, there is an individual appended tube for collection of sample. So that should not be mixed. So each individual are treated with the same restriction enzyme. After that, if they are same species, we'll get same cutting of the restriction enzyme. If it is different species, we'll define different length of the cutting. That means because since there are mutation in different species, sequence are not same. Their restriction sites are in different places, which will give different length in their gel profile. That means you have to run an electrophoresis gel, where we'll confirm the DNA. Again, AFLP, this is another marker. RAPD, randomly amplified polymer, we will select the random primer for PCR. And for that, there are different amplification profile. We will not go in details. VNTR, microsatellite, and SNP. So microsatellite and SNP are very high profile markers, which are used mostly widely for the population genetic studies, where the last part, bioinformatics is helping us tremendously for different aspects like phylogenetic reconstruction, analysis of sequence of the microsatellite. Those can be done only with the help of bioinformatics. Otherwise, it will have some error if we do manually. So if you think about insect, suppose you can see the starting to where how bioinformatics is linked to the DNA extraction to the sequencing. You can see that, for example, we are interested for particular insect, for example. So insect can be we have to isolate the DNA genetic material for each and every insect species, whatever we are where we think that these species are related. Suppose we are interested for phylogenetic analysis or Suppose they are from the same related species. So for that, we have to isolate the DNA for each and every sample we have collected. After extraction of DNA, we must do the PCR for the same gene. That means, for example, cytochrome oxidase subunit 1 for one individual means for other also we have to do same gene, because different genes cannot be compared. Same gene must, that means similar type of genes can be compared. So PCR should be done for same gene for different samples. After that, we have to run the electrophoresis, where we'll get the bands. After getting the bands, using the markers, we'll know that this is the particular gene of interest, because we know the size of the different genes. For example, cytochrome oxidase is around 680 base pair long. So if you use a marker of which give a band of 680 base pair, means that is the cytochrome oxidase subunit 1. So after that, those gels are purified and subjected for sequencing. After once we get the sequence, we can start with the help of bioinformatics. That means how we can, this slide shows how wet lab data is linked to the bioinformatics. That means the work of bioinformatics starts, in case of molecular biology, it starts with the getting from the sequence. That means one must work in the dye lab, wet lab. Wet lab means in our normal laboratory. To start with the dye lab. Dye lab means bioinformatics lab, where no water is involved. Dry lab. That means wet lab data must be generated to work with the bioinformatics in the dry lab. So if it is sequence, what can be done? We can go for phylogenetic analysis. We can go for gene annotation. Whether genes, that means phylogenetic analysis means to study evolutionary relatedness. Gene annotation means we can see which genes are present or which are absent, or where is the location of particular gene. We can go for comparing different genomes. Again, detecting regulatory network or metabolic pathways. Again, proteomics. Proteomics means proteome means total protein produced by a genome, and genome means total genetic material produced by a genome means total genetic complement in an organism cell. Because genetic complement means, for example, human genome means in human we are having 22 pairs of autosome and 1 pair of sex chromosome. That means 22 pairs means each and every from chromosome number 1 to 22 we are having the 2 copies of each. We have 2 copies of chromosome number 1, 2 copies of chromosome number 2. So one set from 1 to 22, that is one set, and it is female, only X chromosome. That means from 1 to 22 and X is called constitute the female genome. And again, from chromosome number 1 to 22, then X, then Y, that means 24 different types of chromosome will constitute the male genome. So likewise, genome means total haploid set of genetic material in an organism cell. Individual cell, how much genetic materials are present, information are present in a cell is called genome. And proteome means total protein expressed by the genome, that is called proteome. And study of proteome is called proteomics. So there might be question how this organism is related to other species. Our new organism is related to other species. For that, we have to get the sequence for the same gene. After that, we have to compare the sequences, which is called sequence alignment. Sorry, you can see here good alignment and bad alignment. You can see mutations are there. In case of suppose you can see 7 and 6, you can see. So in I there are lots of changes. I is divergent from the others, but this is not the case. In case of good alignment, you can see I has the probability to experience a deletion at position 6 or 7. You can see position in position 6. You can see here is a, in 7 there is a C instead of G. In most of the sequences there is a G. G means guanine, but it is having C, cytosine. That means that is called, when G is replaced by C, will come to that is called transversion. So finally, we can go for phylogenetic analysis using those sequence information. That means evolutionary profile. Again, under sequence analysis, we can perform thousands of things. Like we can go for RNA splice site, we can go for ORFs, open reading frames, we can go for amino acid proposition or of the composition of the protein, conserved region. We can see DNA, RNA, DNA to RNA. We can go for DNA sequence to RNA sequence, and we can go for RNA secondary structure prediction also with the help of sequence information. So higher level, we can go for different things. After getting the once we get the sequence, we can go for different types of genomics. Genomics is divided into three different types: functional genomics, taxol genomics, and comparative genomics. So functional genomics deals with the analysis of gene expression, analysis of regulation, analysis of mutation in cancer. Such type of things can be addressed once we get the sequence, genome sequence. Again, taxol genomics deals with the sequence analysis, genome annotation, and comparative genomics. That means once we have the, for example, one of the good example of comparative genomics is that comparative genomics study shows that there are 1.8% sequence difference between human and chimpanzee. Only because of 1.8% sequence difference, human is human, chimpanzee is chimpanzee. Otherwise, chimpanzee will become the human if that only 1.8% will be similar. That means only 1.8% sequence difference between human genome and chimpanzee genome. Again, coming to the proteomics, we have analysis of protein expression, prediction of protein structure. We can go for drug designing. Once we have the protein sequence, from DNA sequence also, we can get protein sequence from protein. We can model the protein, or we have the experimental structure also. We can go for drug designing, because the modern drugs are what? Modern drugs are based on that is called structure-based drug designing. Drugs are based on what? Some targeted to some protein structure. For example, Taxol. Taxol is a drug which is derived from the plant called Taxus polycina. Taxol, and the target of Taxol is that tubulin protein, which is involved in the microtubule formation. The target of Taxol is to block the tubulin protein, because in cancer, what is cancer? Uncontrolled growth of cell. So continuous cell division will take place. So to stop the cell division, tubulin protein will be blocked with the help of Taxol. That means for that we have to understand the structure of tubulin. So once we understood the structure of tubulin, scientists do the research. After that, they found that the Taxol will be the suitable drug which can easily bind to the tubulin protein to block its function. So Taxol will bind to the tubulin protein and it will stop the microtubule formation. So it is a good drug for cancer. Likewise, scientists are looking for SARS coronavirus also for regarding different drug designing, targeting to the different protein, for which the understanding of structure is must. Again, computational evolutionary biology, same thing, phylogeny, measuring of biodiversity and modeling of biological system. At higher level, we can model the system. There is a branch of biological systems biology. Good example, we can see some of the comparative genomic studies. Here you can see the comparison of different genomes, human, mouse. So human genome is 3.2 billion nucleotide base pair, mouse genome is 1.6 billion nucleotide. Gene number is approximately same, although human genome is longer. However, the gene number, that means longer genome means does not indicate that presence of the elephant is larger animal, suppose. Whereas a mouse is smaller animal, suppose. Larger elephant might not have the large number of genes. It might have lesser number of genes than the mouse also. So that is called complexity of the genome. Again, those things can be compared under comparative genomic studies. Different genome you can see in this table, the number of genes. So those can be compared with the help of comparative genomic studies. Likewise, for SARS coronavirus also, scientists are comparing the other coronaviruses, because there are 6 other coronaviruses which are causing disease to the human being. This is the 7th one. SARS coronavirus is the 7th one. So we can compare 7 different coronaviruses and found the differences, where are the similarity, where are the differences. On the basis of that, scientists are planning for different development of the drug or medicines. Again, the concept of database, we can store the huge amount of data in the database. In biology, we are having different types of database. You can see starting from nucleotide sequence database, where all will have the only nucleotide sequence, RNA sequence database, where only RNA sequence will be there, protein sequence databases. That means once we are looking for the nucleotide sequence, we have to go to the nucleotide database. We will not, in structure database, we will not get the nucleotide. That means one must know where is the data actually. So for taxonomy, we should not go to the nucleotide sequence database, we have to go to the taxonomy database. Likewise, there are different databases, where database means the record keeping system. The data of relevant data are stored, or where data in database, information can be stored, information can be retrieved, and information can be updated. So these are the there are 3 nucleotide databases where nucleotide sequences are there, and CBI is there. Again, EBI is there. Then DDBZ. There are 3 primary nucleotide databases. That means where new sequences of DNA or RNA can be deposited, and they exchange their data on regular basis. That means once we, the same example, suppose we can use the HDFC Bank ATM, we can use SBI also. So same thing here. If we are having submitted, if we have submitted on sequence, scientists have submitted on sequence in the NCBI or GenBank, that can be retrieved from other database from Europe. This is in USA, this is in UK, and this is in Asia, Japan. So if you one scientist submit a sequence in the DDBJ, in next week that can be retrieved from the USA also, this database. That means they exchange their data on weekly basis. So that means there are how many sequences of nucleotide of SARS coronavirus means we have to go to these databases. We can search, and these are the authentic source where we will get the actual data for the nucleotide sequence. And scientists can download the sequence and use for bioinformatics also, because these are public domain database. Once it is released in this database, anyone in the world can use without any restriction. So you can see in the this is another database for protein sequence where protein sequence are submitted. That is called UniProt, Universal Protein Knowledge Base. Again, there are some tools called BLAST and FASTA which can be used for searching similarity and homology. That means in forensic science, like forensic science, it can be used. That means if somebody is getting a new sequence in Dhing College campus, suppose one sequence for any nucleotide, suppose DNA sequence, somebody is sequencing. So we must suppose we do not know what is that sequence or what is the organism. So using these tools like FASTA or BLAST, we can perform. After that, we will know what is the exact protein or exact gene of that sequence, and we will know the organism also. We can identify the organism. That means this species is this gene is for this sequence is from this organism. Again, there are different things we can do. Sequence alignment we can do. This is the example of sequence alignment. Again, we can see this is the composition of HTC nucleotide. You can see percentage of HTC can be, and this will give meaning. If a sequence is having higher GC content, G and C, that will give different meaning, because G and C is having 3 different hydrogen bonds. 3 hydrogen bonds. At A and T is having 2 hydrogen bonds. So GC means GC protein means sequence means highly stable. Again, considering amino acids, different percentage of amino acids will give different meaning. For example, higher leucine and leucine-rich protein or higher valine-rich protein means those are highly stable protein molecules. Likewise, we can see size versus pH graph with the help of bioinformatics. We can, again, molecular modeling. In molecular modeling, we can go for modeling of different proteins. There are different methods for protein structure determination. The experimental methods are like X-ray crystallography, NMR, and cryo-electron microscopy. But in the absence of those experimental structure, one can go for computational method like homology modeling, full recognition by 3D, and ab initio modeling. And accuracy is more in case of homology modeling, which can give result up to X-ray accuracy. It has been well established in a conference called as critical assessment of technique in protein structure prediction. Actually, homology modeling, the concept behind homology modeling is that if you are having an sequence, nucleotide protein sequence, but the protein is not having its structure, but it shares some similarity with the existing protein structure. So the existing protein structure of the database can be used as a template or model to build the structure of the new protein. That means once there is a sequence with us, protein sequence with us, but that is not having structure, but that sequence share some similarity with the existing protein structure. That means existing protein structure is having the sequence. That sequence shares similarity with our unknown protein. Then existing protein structure can be used as a template to build a model. That concept is called homology modeling of the protein. So modeling can be done by anyone, but most important thing is the quality of the model, or whether that structure is valid for the scientific community. So for that, there are the model must be passed with some of the parameters, like there is a parameter called Ramachandran plot. In Ramachandran plot, The allowed regions are there in the Ramachandran plot. The more than 90% amino acid must be in the allowed region for a good quality protein structure. And a good quality model generally means there is a software called Q-mean. So Q-mean score around 0 and score below -4 are indicate low quality structure. A lower mole, there is also another software called mole property. Lower mole property score, a LG score is greater than 3, a submax score greater than 0.5. These are some parameters. So those parameters must be fulfilled. Then only we can say that this structure is a good quality protein structure. And there are some well-established university softwares which will validate the protein structure. So Q-mean is software by Swiss Institute of Bioinformatics. Again, ProQTD. Again, this is University of Reading. They are having different softwares, modeful, which are used for structure verification. That means once scientists are modeling the protein structure, that model structure must be verified with the help of some expert or some softwares available in some servers. After verification only, if the parameters are fulfilled, then only we can deposit the structure for the scientific community so that they can use for the betterment of the human being. Again, phylogenetic, coming to the phylogenetic analysis. So phylogeny will answer various questions. Suppose one question is that whether human beings are evolved from the monkey. In this slide, I am showing that. So actually, human beings are not evolved from the monkey. This phylogeny shows that humans, that means monkeys and human beings, are having common ancestor. That means monkey purvapurush and manuhar purvapurush eka asil. But human beings are not evolved from the monkey. So such type of questions can be used from the phylogeny. So phylogeny is the study of evolutionary relatedness among various groups of organisms. Again, there is a branch called phylogenetics, which on the basis of phylogenetic tree, this branch classify the organism. That means whatever classification nowadays we get in the books are based on the molecular phylogeny only. I will go into this slides in details because there are in details. These are different parts in the phylogenetic tree. There are internode, basal node. They are actually for understanding the phylogenetic tree. And there are two types of phylogenetic tree: rooted tree and unrooted tree. In phylogeny, there are various some other concepts. We will not get in detail, go in details because for graduate student or UG student, it will be very difficult to understand. So there are monophyletic, polyphyletic, and paraphyletic concept is there in phylogeny. So how will, from which data will go for phylogeny? So we can, earlier people used morphological data. Different morphological data parameters are collected. After that, there are some metrics are formulated. And nowadays, people are highly dependent on the sequence-based phylogeny. You can see there are two different methods of phylogenetic reconstruction. One is called distance-based method. Second is called character-based method. Two different methods under which there are different other methods. You can see. So most widely used method nowadays is the maximum parsimony and maximum likelihood. These two methods are highly reliable for phylogenetic analysis. So this is the example of a distance matrix for phylogenetic using distance-based method. And in character-based method, each character, there are here in this slide, you can see around 14 different sequences are there. And this, that means in the row, these are different sequences, different taxa, different species in the row. In the column, you can see the position. So each column is considered as a character, where that means you can see first column G, 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 A. That means in the fourth sequence, there is a mutation. G is replaced by A. When a G, guanine is replaced by on adenine, that means purine is replaced by a purine, that is called transition. And when a purine is replaced by a pyrimidine base, that is called transversion. So anyone can do construct phylogenetic tree, but again, there is also some reliability of the phylogenetic tree. That means there is, like we do titration for several times. When we are doing titration in chemistry or water parameter analysis, we repeat the process for 3 to 5 times. Likewise, in phylogenetic tree also, we repeat the process up to 1,000 times, so that we'll get the accuracy that is called bootstrapping. So these are tools for phylogenetic analysis. Mega. So this is one of our paper example of phylogeny based on the, this is a molecular marker called ISSR, interspersed single sequence repeat. So this marker was developed with the help of distance-based method of phylogeny. Again, this phylogeny is using cytochrome B sequence, mitochondrial, completely sequence based. So this is called character-based method phylogeny. Why distance-based method? Because in this phylogeny we are getting different bands. These bands are analyzed with the help of computer softwares, whether they are similar bands or different bands with the help of software. And on the basis of bands, we are preparing the matrix, distance matrix. Only band, no sequencing, no sequence data. So this is called distance-based method. And in this, we are using character-based method. We are using sequence of the nucleotide of cytochrome B for phylogenetic analysis. So in summary of the applications, we can see bioinformatics is having different application: molecular biology, pharmaceutical industry, information technology, database construction, biotechnology, computer-aided drug designing, so on. So now coming to the main part, last part. Sorry. Scientists are using bioinformatics for novel SARS coronavirus, like the popularly known as COVID-19. COVID-19 means CO means corona, V means virus, D means disease, and 19 is the year, COVID-19. So a case study on sequence analysis, phylogeny, protein modeling of the novel coronavirus. That means whatever I have shown in the last slides. Now I'm going to show you the application part, how those concepts of homology modeling, how those concepts of phylogenetic analysis, how those concepts of sequence analysis are applied to the SARS coronavirus. So you can see the scientists around the world are now focusing mostly on, because since experimental data in the laboratory are very poor. Now it is more in the earlier time, it was very difficult to get the information in the experimental laboratory. So people are funding suppose National Science Foundation is one of the most popular organization in US. They are focusing on a project called COVID-19 analysis performed with Galaxy Bioinformatics platform. That is using bioinformatics, they are solving the questions regarding the COVID-19. Again, you can see Texas Advanced Computing Center. They are funding for COVID-19 analysis performed with the Galaxy of Bioinformatics platform. That means bioinformatics is an important subject for not only for this COVID-19, in Human Genome Project also. Actually, Human Genome Project was completed 3 years ahead of the target. That means 2 years ahead. In 2003, it was completed. 1990 to 2003. It was supposed to complete in 2005. Only because of input of bioinformatics, Human Genome Project was possible. Because for analyzing the sequence, for assembly of the sequence, it is very difficult part. Again, in COVID-19 also same case. With the help of computer, with the help of advanced computing, so lots of questions are answered regarding this COVID-19 also. So mostly all scientists are mostly focusing on the construct. Suppose some reputed organization, they are establishing the bioinformatics databases on the COVID-19. Again, sequence comparison databases of COVID-19. Again, phylogenetic analysis database and building of structure models, drug designing. So those 3, 4, 5. These 5 are the promising area. Actually, not promising. There's an ongoing research area of bioinformatics in the field of COVID-19. You can see these are NCBI homepage. You can see National Library of Medicine. This is the actually NCBI, National Center for Biotechnology Information. So they're highly focusing the special database for the COVID-19. This is the specialized database for the COVID-19. On the data, you can see how many nucleotide records are there. So far, 28,843 nucleotide sequences are deposited. This is the data of yesterday evening I have collected. So you can see so far this much of clinical trials are going on. 3,611 clinical trials are going on through all over the world. You can see. So these up to the data are available in the database only. That is called biology. That under biological database concept. That means SARS-CoV-2 databases. Again, you can see this model is a software actually for designing for modeling of the proteins. this Swiss model is having its now database called severe acute respiratory syndrome to SARS coronavirus database for the protein structures. That means whatever protein structures are available so far, they are keeping it as a database record for the scientific community so that they can use and they can get some solution to the ongoing problem. So let us know about some brief introduction to the behind the. I will not go to medical details, but some of the bio-related information regarding sequence or bioinformatics I will go focus. So severe acute respiratory syndrome coronavirus 2, shortly known as SARS-CoV-2, is a positive sense single-stranded RNA coronavirus. That means the genome is RNA genome. RNA is the genetic material, not DNA. So that RNA genome of SARS coronavirus has 29.9 kilobase pair. That means 29,900 nucleotide long, base pair nucleotide long, and it encodes 14 open reading frames for 29 protein. Actually, there are mostly 14 major proteins, but some of the proteins are very complex proteins where around 15 different proteins are combined to form a new protein. It's called polyprotein. So there are 29 different proteins. Again, SARS-CoV-2 is the 7th coronavirus that is known as to cause human disease. What are the earlier coronavirus? You can see previously identified human coronavirus are alpha CoV2, HCoV, NL63, first one. Then again, you can see SCOV229E, second one. Third one, you can see beta coronavirus, SCOV OC43, fourth. Again, fifth one is KU1. Again, you can see SARS-CoV. Again, you can see MERS-CoV, Middle East Respiratory Syndrome coronavirus. So these are the six previously identified coronaviruses, but the new one is the 7th one. That means, although it is novel, actually the World Health Organization, WHO, introduced the name COVID-19 to denote this disease, the epidemics of this disease. They use the COVID-19, but this is not the scientific term. First, they used the term 2019 novel coronavirus. This is also not the scientific term. Actually, the scientific community is addressing it as a SARS coronavirus 2. The International Committee of Taxonomy of Viruses officially designated this virus as SARS coronavirus 2. But whatever name we are using, the COVID-19 is given by World Health Organization to concentrate on these epidemics, and the actual name is SARS coronavirus 2. The scientific community is using. So this is one of the research preprint actually where submitted. These are all in silico proteome analysis of severe acute respiratory syndrome coronavirus 2. So we have actually this is already published in the database, but this research paper is under review. Cold Spring Harbor Laboratory, it is published. So in this paper we have a model using bioinformatics. We have a model out of 29 different proteins, 15 different proteins are not yet experimentally available. Structure are available. So for 15 proteins are model in this protein. Again, this paper is available in European PubMed also. In silico proteome analysis of severe acute respiratory syndrome. So some of the highlight of the coronavirus the genome we can see. This is the genome organization of the SARS coronavirus. You can see. So genome is having like this structure. The genome size is 29.9 kilobase pair. GC% is 38.0% and 11 genes. What are the 11 genes? You can see ORF1AB, ORF1A, then S gene, which is for surface glycoprotein, E is for endoprotein, 7A, ORF7A protein. Likewise, you can see this is the structure. In details, if you see in this table, you can see this is the actual position wise. You can see from nucleotide position 266 to 21,555. ORF1AB, which is the largest polyprotein, is located. Again, you can see 266 to 1Q4683 ORF1AB, 1A protein, polyprotein. That means these two are the most largest protein. And ORF1AB protein is having the 15 different proteins. It is made up of 15 different proteins. You can see the names NSP1, NSP2, NSP4. You can see up to. You can see ORF middle second column. You can see ORF1AB. So 15 different proteins are combining to form a polyprotein that is called ORF1AB polyprotein. Again, there is a structural protein called spike protein. Again, which the location of the genome is 21,563 to 25,384. Again, you can see ORF3A, another protein. 25,393 position of the genome to 26,220. Likewise, last protein is that ORF10. You can see 29,558 to 29,674. That means 29.674, the size of the genome. So these are the different protein. In this slide, I am showing 25 different proteins. And this genome sequence is known as reference genome, which is used by the scientific community all over the world as a reference sequence, because this was the first sequence information submitted to the NCBI from the Wuhan coronavirus. So these are different proteins. We have model using bioinformatics. 15 different proteins are here, which are modeled using the bioinformatics tools and which are verified also as a correct structure and deposited to the database. And these are available to download also. That means one of the application of bioinformatics is the protein modeling of different coronavirus in the absence of their experimental structures. The same approach is done by different other scientists in around the world, but the accuracy of the model varies from structure to structure from various organization. The most accurate structure will be acceptable. Again, you can see there are some of the active site or some of the tunnels under analysis. These are our own analysis. 15 predicted protein structure of the SARS-CoV-2 proteome along with the estimated tunnels. Actually, tunnels are the main points from where the main point of entry or active sites of the protein. Again, you can see these are some of the amino acid composition of the. You can see the green color. You can see those are mostly leucine. That means the high leucine and valine content in E protein means envelope protein, surface glycoprotein, ORF1A polyprotein, ORF1A polyprotein, and high leucine in the isoleucine content in the M protein. Indicates their high structural stability. That when a protein structure there is a high percentage of valine or leucine, those amino acids are highly involved with the structural stability. That means from amino acid composition itself, we can know whether structure is weak or strong or like that. Likewise, this is some of the phylogenetic analysis on the coronavirus. You can see actually not colored, actually same color. In the middle, you can see its SARS coronavirus resembles with one of the bad coronavirus from China. These are different coronaviruses from different species. I have another phylogeny I will show you in details. So these are some submission of the structure. You can see computational model structure of SARS coronavirus to spike glycoprotein submitted to the model archive. This was submitted on 3rd June 2020. So this is one research paper which is already accepted. This is another work. This was another work. First was this another work. This is another research paper which has already been accepted and it is going to be published within this week. And this is under in Biomed Research International Journal, Impact Factor 2.7. So in this research, what we have done, sequence analysis and structure prediction of SARS-CoV-2 accessory protein 9B and ORF14. Evolutionary analysis indicates close relatedness to bad coronavirus. So in this paper, what we have done, 2 proteins are there. Most of the genome sequences are going on around the world, but most sequencing project miss these 2 proteins. Although there are 29 proteins of coronaviruses, but only 25 are mostly. That means the genome sequence for encoding 25 proteins are mostly sequenced, and around 4 proteins are missing in the data. Only 1 or 2 genome sequence reported these 2 proteins. That means very less work was there on these 2 proteins. No structure is available, no experimental structure in the database. So using this sequence from the database information, we have analyzed this structure using bioinformatics. So what we have done, the concept behind this work is like this. We have downloaded the sequence, UniProt reviewed amino acid sequence of the SARS-CoV-2 and ORF14 was downloaded from the database, UniProt database. So we have done 3 different types of analysis: A, comparative and ab initio modeling; B, proteomic analysis and functional annotation; C, molecular phylogenetic analysis. That means in this research paper, 3 different approaches are done One is protein modeling, secondly proteomic analysis, and thirdly molecular phylogenetic analysis. So under this, we have done 2 different types of modeling. We will not go in details. So one is comparative modeling, second is called ab initio modeling. 2 different modeling methods are used for model the protein. Under B, proteomic analysis, what we have done physicochemical parameter analysis. So how much of what is the aliphatic index, what is the isolytic point, what is the molecular formula, like that analysis. Again, functional annotation, then prediction of biochemical function, biomolecular channels and tunnels analysis, computation of the cavities of the protein. Again, molecular phylogeny of the 2 protein sequence based phylogeny. Again, we have also done whole genome sequence phylogeny of the coronavirus. We have compared both the data for phylogeny. So you can see some of the results here. This is the blast result in NCBI. We can see 89% query coverage, 89% sequence is covered. That means the ORF9P protein shared 89% identity with the existing protein structure in the database. That means no 100%. 100% means it is already available. The structure is already available. So since it is 89%, that means no percentage identity is 70% only. 30% is different. So 70% identity means the structure is not yet available in the database. So since it is 70%, good identity is there, so we can use for comparative modeling. That means a protein structure having more than 40% identity when you use for modeling, that will be a good quality model. Then less than 25%, we should go for ab initio modeling. Here it is 31%. It is better to go for ab initio modeling since it's less than 40%. So one protein is modeled using comparative modeling procedure, and second is using ab initio modeling procedure. Here you can see some of the parameter from different, not only these 2 proteins, for other coronavirus also it is here. Human SARS coronavirus to bat coronavirus. You can see length, molecular weight, isoelectric point, chemical formula, instability index. So one point here I will focus is that the instability index value, once it is more than 40, then that protein is said to be stable. When it is, instability index value less than 40 means it is stable. It is more than 40 means it is unstable. Protein is unstable. Likewise, aliphatic index, higher aliphatic index means proteins are having high thermostability. Here you can see these 2 proteins are highly thermostable. That means high heat also it will not denature. Again, GRA BY means grand average hydrophobicity. Hydrophobicity means whether the protein is indicative means the proteins are hydrophilic. You can see the hydrophilic nature of human SARS coronavirus 2, ORF9P protein. Likewise, other protein you can see positive is there. That means that is hydrophilic. No, positive means hydrophobic, negative means hydrophilic. GRA B value, negative means it is hydrophilic. Positive means it is hydrophobic. So likewise, you can interpret the data. You can see here. Again, in this protein, you can see leucine is higher and pheline is higher. So these proteins are highly stable protein. High structural stability is there for these 2 proteins. Likewise, you can see this is the protein structure. Here is the in the second diagram. Here some analysis on the active site. In this protein, second protein also some of the analysis. So this is the phylogeny. You can see this is the whole genome phylogeny. That means whole genome sequence was considered for constructing the phylogeny. In the middle, you can see. In the middle means from bottom 1, 2, 3, 4, 5. You can see in the middle here the new coronavirus resembles with the bat coronavirus RATG complete genome, which was isolated from China. Again, it resembles with some pangolin coronavirus. That means new coronavirus has resemblance with the pangolin and bat coronaviruses. That means the earlier old coronavirus also that resembles with the some 2004-2006 bat coronavirus. That both the time, that means in the both the time there is a possibility of transmission from bat only. The earlier also whatever coronavirus was there, that is having similarity with the bat. This time also it is having some similarity, higher similarity with the bat. That means there is a possibility of both the time, earlier also, this time also. That earlier means SARS coronavirus, this time means SARS coronavirus 2. So, but the time there is a possibility that bats are transmitting this disease. So, phylogenetic analysis of the protein revealed that close evolutionary relationship between the newly emerged human SARS coronavirus 2 and bat SARS coronavirus. Again, whole genome phylogeny also shows that it is may have originated in bat, undergo repeat evolution in bat, and pangolin may likely be amplifying host. The presence of a large number of tunnels in 9B protein indicates high reactivity. So likewise, we have interpreted the data. This is for protein phylogeny. Secondary structure of the protein. Again, verification of the protein with the software. These are verification reports. So these are our own. Again, some other scientists, what they are doing, they are also doing some potential antiviral activity of the approved. That means drug designing. This is from Assam only. These authors are from Assam. So they are also trying the drug designing approach for targeting one particular protein or the protein. This is another application of the bioinformatics for SARS coronavirus. Again, you can see advanced bioinformatics rapidly identifying existing therapeutic for patients with coronavirus disease 19. You can see lots of research paper in this field. Again, you can see computers and viral disease preliminary bioinformatics studies on the design of a synthetic vaccine and preventive predominant antagonist against the SARS coronavirus. So there are lots of research is going on around the world regarding the SARS coronavirus, mostly phylogeny, drug designing, and protein modeling, and so on. So thank you. If you have any query, I will try to address. And for actually beginner, it is very difficult to understand also. But since the level of participants are starting from undergraduate to research level, so I'm focusing both basic and advanced things. Thank you.
Thank you, Dr. Pura. 3 questions for you. Hello.
Yes. Is there any question?
Hello.
Yes.
First question is Dr. Punima Daspora. She asked you, can you please provide us the technique, if possible, essay used in identification of organism at species level?
Yes. For identification of organism at species level, we can use, as I told, we have to pass. Can I show the slides? I'll show the slides. It will be easier for understand. I'll show the slides. It will be easier to understand. I'll come to that. Okay, for identification of organism at species level, we have to it will be easier to understand, otherwise difficult. You can see this is the slide I'm showing actually. For this is the actual technique for identification of organism at species level. For the organism, first of all, we have to select, we have to isolate the DNA of that organism, what we are going to identify. We can, for if it is a fish, we will not kill the fish. We can simply have a scale of the fish only. Bakoli to hole masor, kotha nai. Jikono body part, chuli hole to animal jodi hoy. Non-invasive technique kori we can take the sample. So after taking the sample, you have to isolate the DNA. Our DNA isolate kori, we have to select the appropriate marker, whether we are interested for cytochrome or not. So marker to select kori. After that, we have to go for PCR for the particular marker. Then we have to get the sequence. Then once we get the sequence, we can go for, there are some tools called BLAST and FASTA. Using BLAST and FASTA, we can compare the sequence with the existing database sequence. Likewise, we can identify the organism, new organism, using BLAST and FASTA. That means we must have the sequence information. We must sequence the organism. DNA sequence like bioinformatics. Sequence takle parim.
Thank you. Next question is, ask our student, full form of SARS CoV-2.
Already called column full form of SARS-CoV-2 column first slide. মই দেখুৱাই দিছো বস্তুটো এটা মই এলা খুদিব বুলি জানি মই লাইনৰ কথা বেছিকৈ দিছো মই এক মিনিট ৰখক মই দেখুৱাই আছো স্লাইডখন দেখি আছে চাগে মই যত আৰম্ভ কৰিছিল তাতে চাই লওক এইখন চাই লওক এইটো SARS মানে সিভিয়াৰ একিউট ৰেসপিৰেটৰি সিনড্ৰম SARS মানে আৰু CoV-2 মানে লিখা আছে ধুনিয়াকৈ CO মানে আছে করোনা V মানে ভাইরাস VI মানে ভাইরাস D মানে ডিজিজ আৰু 19 মানে ইয়াৰ SARS-CoV-2 যদি হয় SARS coronavirus 2 severe acute respiratory syndrome, coronavirus 2.
Yes. Another question is asked for you, Jaharuddin Ahmed. Can single cell RNA sequence determine which cell types are infected by COVID-19?
Yes, actually, whatever protein will be coded by the genome, RNA genome. That means the RNA is the genetic material in the coronavirus. So the genetic material itself will contain the information which part will. That means it is already determined which part of the genome is responsible for which part. That means as the genome I have shown here in the slide, different parts are already responsible for different protein. You can see here somewhere there is a spike protein. Spike protein is there, S protein, surface protein, E protein is there, envelope protein is there. That means already 26,245 to 26472 is responsible for M protein, membrane protein. That means each and every part of the genome is responsible for particular protein. That means the protein are performing the function. So it is already determined. You can see this slide itself, it is self-explanatory. So you can see that each and every part of the genome is responsible for particular protein and performing the function. Mainly spike protein, one of the main protein is the spike protein in case of coronavirus. So that protein is responsible for forming spikes. Or likewise, later protein is there. You can see N protein, nucleocapsid phosphoprotein. That is also responsible. So it is already determined which protein is responsible for what. Spike protein will first bind, like that. Okay. The RNA genome itself determined. In the genome information sequence itself determines what will be the function of the genome.
Thank you. Thank you, Dr. Chittaranjan Borua, for your precious and informative presentation. I hope our students and all participants are benefited from this information. Now I request Dr. Ujjanta Das offer vote of thanks for all. Thank you.
Thank you, Luna Madam. Respected Principal, Dhing College, respected resource person of the webinar, and Honorable Coordinator and Convener of the webinar. I, Dr. Ujjanta Das, on behalf of the webinar committee, would like to offer my sincere gratitude to Dr. Biman Hazarika Sir, Principal Dhing College, who have supported us to his best to make this webinar a successful one. I would also like to shower my sincere gratitude and thanks to our resource person, Dr. Chittaranjan Borua, Assistant Professor, Department of Zoology, Darang College, who has given us a brief knowledge on application of bioinformatics, a case study on sequence analysis, phylogeny, and protein modeling of novel coronavirus, which I believe to be an important topic in this present situation. I also shower my thanks to the respected coordinator, Dr. Luna Fukan, head of the Department of Zoology, convener, Dr. P. Das Bora, Zoology Department, and colleagues for their all-round support to materialize this webinar. I would also like to thank to the respected participants for their participation and active interactions, without whom the webinar would not have been successful. Lastly, I would like to thank to the technical staff who have supported us to conduct this webinar successfully. Thank you, sir.